This is an overview for Chapter 2 in your Colville and Bassert Clinical Anatomy and Physiology for Vet Techs. And this chapter is on the chemical basis for life. So a lot of interesting facts in here about how your cells are made up and what kind of chemical reactions are going on in your body. Learning objectives for today are to list the characteristics of each of the subatomic particles, differentiate between a molecule and a compound, list and describe the types of chemical bonds that may form between elements, give the general equations for the synthesis, decomposition, and exchange reactions, differentiate between organic and inorganic compounds, and between hydrophobic and hydrophilic molecules. We're going to list the unique properties of the water molecule, differentiate between acids and bases, describe the actions of a buffer system, list the components of carbohydrates, lipids, proteins, and nucleic acids, and the functions of body proteins, as well as the actions of enzymes. So let's start with the basics. Everything is matter. So matter is defined as something that takes up space and has mass. So that's everything. Elements are things that cannot be divided by ordinary chemical processes into another substance. So the very basic elements. There are 92 that occur in nature. You can see them below on the periodic table. Some examples would be gold, oxygen, chlorine, etc. These chemical symbols that you see on the periodic table are derived from the name in English, Latin, or Greek. So O is oxygen, AU is gold, and these can be found on that periodic table by order of atomic weight. Only 4 of these are found to make up 96% of the matter in living organisms, and that would be hydrogen, carbon, nitrogen, and oxygen. And those are identified in red on the periodic table there. There are also macrominerals, which are identified in blue, and that's sodium, magnesium, phosphorus, sulfur, chloride, potassium, and calcium. And we call those macrominerals because they are found at greater concentrations within the body. So they're used for more chemical reactions within the body than are these other minerals, which are called microminerals, and those are identified in yellow. So boron, fluoride, aluminum, silicone, they're just iron, cobalt, copper, zinc. Those are the yellow ones that you see below there. Table 2.1 in your book has more information about the elements and how they're used in the body, so take a minute to look at that. Atoms are the smallest unit of the element, and that each atom is unique to its element. These atoms are made up of smaller subatoms, and those are the protons, neutrons, and electrons. Protons and neutrons are the heaviest. They actually make up the mass. So a proton and a neutron together makes 1 atomic mass. And they're grouped together in the center, and that's the atomic nucleus. So proton and a neutron together determine the atomic weight of the element, which you can see them lined up on the periodic table based on that weight. Electrons, on the other hand, are tiny and constantly moving, so they're not contributing to the overall mass. Protons are positive, neutrons have no charge or they're neutral, and electrons are negative. Protons, the number of protons equals the number of electrons, so that there is a neutral net charge. And there are 2 models, 2 ways that we can look at these atoms and how the protons, neutrons, and electrons are arranged. There's the planetary model and then the orbital model. The orbital model is actually more accurate because the electrons move into and out of the sphere, the nucleus, and the influence of the nucleus. So there are times when those electrons are kept within or closer to the nucleus, and times when they're probably more able to be shared with other elements. So here's an example of carbon. Carbon, if we were trying to decide its atomic weight, we would look at how many protons and neutrons there are. There are 6 protons, 6 neutrons, and 6 electrons. So what is the atomic weight? It's 6. Now let's go back to the atomic or the periodic table, make sure we're correct. And here is carbon. and its atomic weight is over here, and that is a 6, and it's the 6th element on the table. So 1, 2, 3, 4, 5, 6. So it makes sense. Elements can only change numbers of protons by extreme means. So the protons are the positive charge that are within the nucleus. And it takes a nuclear reaction to change that number. And if it does change the number, then they become another element because they gain or lose mass. Elements that lose or gain neutrons are referred to as isotopes. So they're like the element, but they are slightly different from the element. But elements can lose or gain electrons more easily. And if they do, they're called ions. And they're either positively, meaning they've lost electrons, or negatively, meaning they've gained electrons, charged. The area where the electrons are positioned is within that electron shell. And you can see these shells on the carbon element here. These are considered shells. Now, within the first shell, there are 2 spots for electrons. So this first shell for carbon is completely full. In the second shell, there are 8 potential spots for electrons. And in this carbon molecule, we are missing 4 electrons. So we have 1, 2, 3, 4, but there's space for 4 more. The area where the electrons are positioned as the electron shell, like I said, have a certain number where there's space. And the electrons that are farther from the nucleus actually have a higher energy. Now think about that. It has to move faster to move around that orbit. Figure 2.8 in your book is a really good figure to look at to understand what full and empty shells mean. Atoms are most stable when the electron shells are completely full and they become inert. For instance, helium, which is the second element on the table, has 2 electrons, and those electrons completely fill that first shell. Neon, which is 10 on the table, has 2 electrons in the first shell and 8 electrons on the second shell. So it has 10 altogether. That means it's completely full shells. They are called inert gases. It's like you after Thanksgiving dinner. If you are completely full, you just want to sit on the couch. So you can think of it that way. When atoms don't have full shells, then they try to find electrons to complete them, to complete their shells. So they'll form bonds between atoms. So hydrogen, carbon, nitrogen, and oxygen don't have full shells, so they want to form bonds with each other. What's the difference between a molecule and a compound? So anytime an atom comes together and shares electrons, that's called a chemical bond, and that's called a molecule. If they're the same atoms, that's the molecule of the element. So O2 is 2 oxygens sharing electrons, that is a molecule. If they are different atoms coming together, then it's called a compound. Carbon and oxygen coming together is a compound. So the process of combining is chemical bonding, and it just refers to sharing of electrons. Here's a dimension or a diagram of 2 oxygen atoms coming together and sharing those electrons. So this has 6 electrons in the outer shell. It wants 8, so it meets up with this other oxygen, and they share 2 of their electrons. So together, they each have 8 electrons in the outer shell. And with CO2, we have carbon, which has 6 electrons. It wants 8. So it shares with 2 oxygens who also want 8 in their outer shell. So it works out for them. So that we have 3-dimensional models, we have a line drawing, and we have a planetary model that we can show and describe how these compounds get together or how these molecules get together. All right, there are 3 types of chemical bonds. There's covalent, ionic, and hydrogen. And you probably have guessed it, but hydrogen refers to any bond that occurs with hydrogen. And it's actually a mixture of the covalent bond and the ionic bond. Covalent bonds, the atoms share electrons. So like oxygen and carbon dioxide that we just saw, they're going from the outer shell of one to the other, and they're in constant movement with each other. Oxygen and carbon dioxide, that was one example. Methane or carbon with 4 hydrogens sharing, that is a covalent bond. That means they're co-parenting. You can have a single, a double, and triple covalent bonds, which can be formed. It just depends on the number of electrons that they share. Dihydride oxide is also called water. It has an arrangement wherein the hydrogen is actually slightly positive because it's constantly sharing its electron, so it makes it constantly positive. The oxygen in that group has a slightly negative charge when they're bound together, so it's like a battery. It becomes a polar molecule where the hydrogen is slightly positive and the oxygen is slightly negative. That means you kind of have a battery situation where you have a positive end and a negative end. And that is important because hydrogen and oxygen, when it's combined in water, tends to want to become more stable, so they join with other molecules in order to create a stability. Ionic bonds are when electrons are actually transferred from one atom to another. So it just goes ahead and takes it. It's either in those with fewer than 2 electrons in their outer shells, or with those that have nearly full outer shells. So they're almost there, and they just want to get that last piece of the pie, and they'll just grab it. So sodium chloride is one that is formed through an ionic bond. They're drawn together by electrical charges, and that's called electrostatic attraction. Sodium has 11 electrons, and it just has 1 in the 3rd shell. So it's one really lonely electron in the 3rd shell. It wants a lot more electrons around it. So it desperately wants to lose that electron so it can just become stable. And when it does, it becomes relatively positive. So sodium losing that electron, which it wants to do constantly, is called sodium plus, and that's a cation. Chloride has 17 electrons with 7 in the 3rd shell. So it's almost completely full, and it really, really wants another electron. And in order to gain that electron to become stable, it becomes relatively negative. So it is chloride negative, and it's considered an anion. So a cation wants to get with an anion, and that's an ionic bond. Hydrogen bonds is a combination of the previous two, like I said, and it is a very specific type of weak ionic bond. It's weaker than the other bonds. It's a bond between the hydrogen atom that is already covalently bonded to something else, to another molecule, and to oppositely charged particles. When it is then covalently bonded, it has a weak positive charge because hydrogen wants to donate that extra electron it has to another atom. So it's attracted electrostatically or ionically to a negatively charged particle. So it forms, there's a bond that forms between molecules. Like water will bond with water, but on the opposite, so positive side will bond with the negative side of another water molecule, and that helps to stabilize the solution. It can also form between parts of the same molecule, and this happens within proteins and DNA to add structure. And that's why having protein within your body and eating protein is very important, because we need that protein to develop structure in our cells at the cellular level, all the way up to the tissue and organ level. And it's the hydrogen bonds that help to create that. There are 3 types of chemical reactions, and that just relates to the formation and breaking of those chemical bonds. So think back to chemistry and those chemical equations that you probably really struggled with. I'm going to try to make it a little bit easier here because we're just talking about a few things within the body. X plus Y, X chemical plus Y chemical, or X element plus Y element equals Z. And that's our chemical equation. So thinking about these chemical reactions in terms of those equations, let's think about synthesis. Synthesis is really simple. X molecule or X element plus Y element equals XY, 2 together. Oxygen plus oxygen equals O2. That's a molecule. And in the body, simple molecules are absorbed through digestion to join larger molecules that are needed by cells for life processes. So this happens through synthesis. There's also decomposition, where you have that molecule XY, and it breaks down into 2 different parts, X plus Y. For instance, water. You have two waters that will break into two H2s and an O2. And those numbers all equal the same thing. So there's, in essence, there are 4 hydrogens and 2 oxygens when you're finished with this decomposition. An exchange would be when we have a molecule or a compound, WX plus YZ, a different compound, and you add them together and you end up with an exchange where you have WY and XZ. That should say XZ, not XY. So for instance, sodium bicarbonate, baking soda, plus acid, HCl. This is where we have a base and an acid added together. What do we end up with? We end up with sodium chloride or salt plus water plus carbon dioxide. So in the body, if we're using sodium bicarbonate, which we can use for digestion, say we have an animal that has increased stomach acid, and we give them a little bit of bicarbonate, it will mix with the hydrochloric acid in the stomach. And what does it make? It makes salt, water, and carbon dioxide. So what do you think they're going to do? They've got a little gas, bubbles. They may belch if they are given too much of it. So all 3 of these chemical reactions, when they happen, require or release energy. Synthesis reactions require energy. In order to put these 2 molecules together, these 2 elements together to make a molecule, they need energy to do that. Decomposition reactions release energy. So if we're taking a compound and we decompose it, it releases energy. Exchange reactions, there's no net energy change. So it's 1 for 1. You get an equal reaction. You use a little synthesis, you get a little decomposition. So it's equal. So there are factors which can influence these chemical reactions, and these factors are as follows. There's the availability or the concentration of reactants. So if you have more of a product, you're going to get more of a reaction. Temperature influences the rate of reaction. So an increased temperature means you have increased molecule movement, and that increases the chance that the molecules are going to meet, and also the velocity at which they meet. And then also activation energy. That's the energy that's required for a reaction to occur. If you don't have energy, then chemical reactions cannot occur. Synthesis reactions in particular. Some reactions actually require a catalyst. And for our purposes, we're talking about the body, these are special proteins that hold reactants together so that they can interact. And we call those enzymes. We need both organic and inorganic chemistry to happen in our body to keep us alive. Organic chemistry refers to carbohydrates, lipids, proteins, nucleic acids. They're mostly covalently bonded, and they contain hydrocarbon chains that have a functional group. So they all kind of look the same, except they have this one little side chain that tells this compound what it's supposed to do. We also have inorganic chemistry. So water, how salts interact, how acids and bases interact. Water is the universal solvent. The solutes that things that we call solutes are chemicals that are added to the water, compounds that are added to the water. A solution is water plus that chemical. Water molecules surround that solute or the chemicals, and they form a layer of water. And it's either with the negatively charged ends of the water that are attracted to the positively charged molecules, and these are hydrophilic. They like water. And this would be like a solution like Kool-Aid. Kool-Aid is surrounded by the water, and it is dissolved within the water. Or it could be the positive ends of the water blanket those negatively charged particles. So they're actually hydrophobic. They push it away. That would be like oil and water. Water is an ideal transport molecule. Think about blood because it's made up of water. Water also has a high heat capacity. It absorbs energy, which is given as heat in other reactions. And it also has high heat of vaporization. It remains liquid through a wide range of temperatures. So if we get a high temperature in our body, we don't have, we can have issues, but our blood isn't vaporizing because it's actually made of water. Water is also used for lubrication throughout the body. Salts are used in the body as well. These are mineral compounds that have ionic bonds, and when they're added to water, they immediately ionize, meaning they divide into their separate ions. Electrolytes are salts in their ionic forms. What are some examples of salts? Sodium chloride, potassium chloride, magnesium phosphate, those are all salts. And examples of electrolytes: sodium, potassium, chloride, calcium. Acids and bases. Acids are ionically bonded substances that release hydrogen ions when they're added to water. So they're hydrogen donors or proton donors. Bases, which we also call alkaline, are ionically bonded compounds that release a hydroxyl ion, or OH negative, when combined with water. They are protein acceptors. They will take a hydrogen ion or the proton. So they are also electrolytes. They transmit electricity when they ionize in water. There's something called neutralization or buffering that occurs when an acid is added to a water that already contains a base. I want you to understand the pH scale and what makes that pH higher or lower, and why we call a higher pH basic and a lower pH acid. That's in figure 223 in your book. If you take a look at it, it'll give you a much better understanding of this. Acidity and alkalinity are measured from 1, like I said, most acidic, to 14, most basic. And pH of 7 is neutral, like water. Buffers are weak acids and bases that don't completely ionize in water, so they work to buffer the solution where chemical reaction solutions can take place. So a pH of 7 is what we're looking for when we're talking about buffers. The most common buffer system within the body is carbonic acid and bicarbonate, which can further break down to carbonate and then back again. So if we take bicarbonate, H2CO3, it will break down into a hydrogen proton and carbonic acid, HCO3, which can further break down into water and carbon dioxide, or it can actually form and bond together to make get an ionic bond that makes water and carbon dioxide. And that carbon dioxide can then be removed through respiration, or it could go back into making a hydrogen proton with carbonic acid and then go back into bicarbonate again. Organic molecules are things like carbohydrates, lipids, and proteins. Carbohydrates are used for energy, storage of energy, and cellular structure. For example, sugar, starch, and cellulose. Organic molecules are composed of carbon, hydrogen, twice the hydrogen as the carbon, and oxygen. So we write it as C, H2, and O. Hydrated carbon or water containing carbon is how we refer to it. Figure 225 will actually explain each of these structures to you and how we're writing them down. Now, when we think about organic chemistry, often we'll think about it as a hexagon, and this is the ring structure that the carbon makes when it forms a molecule or a compound together. There are several different types of carbohydrates. We start with simple sugars. They're also called monosaccharides. They are 3 to 7 carbon atoms in a chain or a ring. Glucose, for instance, has 6 carbon, 12, always twice the number of carbon. You'll see the hydrogen, so 12 hydrogen and 6 oxygen. Glucose is the primary fuel of the body, and it's considered a hexose because it's a hexagon shape. Glucose is stored as glycogen in the liver for use later as energy. Fructose is also a hexose. It's the same formula as glucose, but the atoms are arranged differently. it's actually converted to glucose in the body. What is fructose? It's fruit sugar. Ribose is a pentose, and that is a 5-carbon sugar. It makes up the backbone of DNA and RNA. So those are our simple sugars, glucose, fructose, and ribose. Disaccharides have 2 monosaccharides joined through a synthesis reaction. Remember, synthesis reaction requires energy. So water is created during this reaction, and it's extracted from the saccharides, so it is called dehydration synthesis. For example, if we join glucose plus fructose and remove water, that makes sucrose, which is table sugar. Polysaccharides are many monosaccharides joined by dehydration synthesis, and it is a structural or fuel storage function. So glycogen and cellulose are polysaccharides. Glycoproteins are a mixture. It's a macromolecule, a big molecule of a carbohydrate joined to a protein. And it helps with adhesion of cell to cell, and so cells joining each other, and recognition and transport of molecules to be transported into cells. So we need glycoproteins or sugar proteins in order to help our cells work together. There are 3 different carbohydrate reactions that occur in the body. Anabolism is when the cells use synthesis reactions to build molecules that they need to function. We talk about anabolic steroids, and this is a good way for you to remember what is happening. Anabolism builds. If we talk about anabolic steroids, those things that are illegal for athletes to have, there are also anabolic steroids that we give to our animals to help to grow meat. But if you think of these people with anabolic steroids on board, they have major muscle mass. So anabolism is when the cells use synthesis reaction to build molecules they need to function. Hydrolysis is when compounds are decomposed into individual structures by water. So when you add water to sucrose, you get glucose and fructose. Decomposition through this hydrolysis yields energy, which can be used to fuel the reactions in the cell. So you need water for energy. Catabolism is when we have decomposition of nutrients. And we talk about catabolism when we're talking about animals that have cancer and develop cachexia. Cachexia is when you see a wasting away, and that's due to the decomposition of nutrients. So here is the formula that we could write out, talking about how glucose plus fructose through dehydration synthesis, yielding water, will give you a disaccharide called sucrose, which is table sugar. Or you can take table sugar, add water, and you will divide glucose and fructose. And you can go back and forth there. Lipids. Lipids are broken up into 4 categories: neutral fats, phospholipids, steroids, and eicosanoids. They contain carbon, hydrogen, and oxygen, just like sugars, but sometimes phosphorus. But these are lower amounts of oxygen than you would see with carbohydrates. Neutral fats are called triglycerides, or just simply fats, and they contain 3 fatty acids and a glycerol. Glycerol is a modified 3-carbon simple sugar. This fatty acid is a chain of carbon atoms with 1 to 2 hydrogen atoms attached by a single or double bond. And this is where we get into saturated fatty acids versus unsaturated fatty acids. And I'm sure you've heard of these because some are considered healthy for you and some are not. Saturated fatty acids, all of the bonds within the chain are single bonds. All the carbons are full of hydrogen atoms, so they're completely saturated with hydrogen atoms. That we find in animal fats like butter and lard. Unsaturated fatty acids, we have some double bonds between the carbon and the hydrogen, so they're incompletely sharing. They have a covalent bond between the carbon and the hydrogen could break apart and or join something else. And those are unsaturated fatty acids. They're not completely saturated or full. And those are from plant origin, like corn oil and olive oil. And those are the fatty acids that we consider more healthy. and important for your body to have. Glycerol forms that backbone. It looks like an E, and then you have two fatty acids connected to that glycerol. And it's joined by dehydration synthesis, and three water molecules are produced by the formation of this triglyceride. When triglycerides are decomposed, water is then consumed. They're decomposed by hydrolysis, and then water is consumed. And triglycerides, when decomposed, are used for energy. So fats are used for energy, and we do need fats and fat-filled cells to pad vital organs from trauma and act as insulation to help maintain body temperature. Neutral fats are hydrophobic, and they do not mix with water. Phospholipids are similar to triglycerides with glycerol backbone, but they have two fatty acids and a phosphate group attached to the nitrogen-containing compound in each direction. The phosphate group is hydrophilic and polar, and the fatty side is hydrophobic and nonpolar. So it's the main component of cell membranes, and it helps to form that myelin sheath of nerve cells. Those phospholipids joining up together, lining up in two layers, make that lipid bilayer when they are placed in water. Very important as a part of the cell membrane. Steroids are a formation of 4 interlocking hydrocarbon rings. They are hydrophobic, nonpolar, they have very little oxygen within them, and they have attached to them very unique functional groups that change the steroid. For example, basic cholesterol ring is synthesized from acetyl coenzyme A, and it's used in the formation of bile salts, which digest fats. It's used by adrenal glands, testes, and ovaries to make cortisone, estrogen, progesterone, and testosterone. So it's really, cholesterol is actually very important in our body. We just want to make sure we don't have too much of a good thing. Eicosanoids are a 20-carbon fatty acid ring structure, plus a ring structure, and they mediate complex chemical processes. For example, eicosanoids include prostaglandins, which mediate inflammation, thromboxane, which mediates platelet function, and leukotrienes, which mediates bronchoconstriction and increased mucus. Proteins are the most abundant organic molecules in the body with the widest variety of functions. They are part of cell structure, structural body tissues, controlling chemical reactions, regulating growth, and defending the body from invaders. They catalyze or speed up all reactions in the body and transport ions and other molecules in and out of cells. They're made up of carbon, oxygen, hydrogen, and nitrogen. Some may contain sulfur, iron, or phosphorus, and the proteins are made up of amino acids that are linked together. Now, the sequence of how they're linked together defines the function of the protein. So think of a protein as a sentence, and the amino acids are words in that sentence. Now, if you change the way the words are located in the sentence, it could come up with a completely different meaning. So we can think of that as defining the sequence, defining the function. There are 20 different amino acids in the body, and they all have the same basic structure. You have a central carbon attached to a hydrogen atom. Then you have an amino group, which is the H2N or NH2, and you have your carboxyl group, which is COOH. Then there's always this unique side chain designated by R. There are infinite possibilities and combinations, and all of these amino acids are ordered by the DNA, and they're linked by dehydration synthesis. So that means we lose a water when we link them. Carboxyls link to other amino acids by a peptide bond. We can have a dipeptide bond, which is 2, tripeptide bond, which is 3 amino acids. 10 plus amino acids is a polypeptide, and 100 plus amino acids is a protein. So we're just talking about smaller, bigger, biggest words. Table 2-3 has the functions of the various proteins that you will find in your body. Protein structure, the shape of proteins determine their function. There are fibrous proteins that we consider collagen. They're long and firm, and they add strength to tissues. Globular proteins, or immunoglobulins, are what make up antibodies, and these are proteins that have a specific shape to join like a puzzle piece to a very specific foreign protein or antigen that invades the body. There are 4 levels of structure. There's the primary structure, which is simply the sequence and number of amino acids that link to form the peptide chain. So it's the sentence written out for you in a straight line. There's the secondary structure where you have a natural bend, parts of the peptide chain, kind of in a 3D structure. So it's a sentence that is written on a ball and it bends. And those bends are stabilized with a weak hydrogen bond, so they're not going to continue to twist and bend. There is this tertiary structure, which is the basic shape of the protein. There can be an alpha helix shape, like a spring or a slinky, and that's what we see with DNA, or it could be a beta pleated shape, like an accordion. There's also a quaternary structure, where 2 or more proteins form a complex macromolecule. The protein is actually folded on itself to create an inner hydrophobic amino acid layer with hydrophilic amino acids on the outside. And these are also held by hydrogen bonds as well as some covalent bonds like disulfide bonds. And it joins with another protein to stabilize the shape. So there's 2 proteins together making a macromolecule. So structural proteins are stable, they're rigid, and they're water insoluble. They have their hydrophobic on the outside. These are long, stringy, fibrous proteins, and some examples are collagen. These are the main protein within connective tissues. There are functional proteins which are water soluble. They have to be water soluble to enact a chemical reaction. They are flexible, they're 3-dimensional, and they can change under different circumstances. These are our globular proteins, and they're highly chemically active. For instance, hemoglobin, antibodies, protein-based hormones, and enzymes. So how do enzymes work? Well, enzymes catalyze chemical reactions without being destroyed or altered. They are specific to the reaction and the substrates, so the substances they act on, and it's determined by shape, change, hydrophilic and hydrophobic properties. There's also a lock and key. Enzymes will fit its substrates very exactly, and it's unaltered. And enzymatic reactions occur in a series. So each product of the substrate is the substrate of the next reaction. So, for instance, you will have one reaction happening, which the product of that will create another reaction, and the product of that will create another reaction. And reactions will often occur along lipid membranes where the enzymes actually live. For example, the Krebs cycle, remember the Krebs cycle, with electron transport system for the production of ATP molecules. Those are enzymes that create that. Nucleic acids are the largest molecule in the body, and they're composed of carbon, oxygen, hydrogen, nitrogen, and phosphate. DNA is deoxyribonucleic acid, and it is the blueprint. It's in the nucleus and the mitochondria of the cells. It contains all the instructions to build proteins. Instructions are coded in segments of DNA called genes, and there are 2 parallel strands of ACGT, which is adenine, cytosine, guanine, and tyrosine, and those hydrogen bonds are between those nitrogen bases, so it creates that double helix shape. A only bonds with T, and G only bonds with C, and the order of this bonding is unique for individuals. RNA, or ribonucleic acid, is the scanner or the fax or the printer. It transfers instructions from the nucleus, from the DNA, to the cytoplasm. It builds proteins that catalyze all reactions performed in the cell. It's only one strand. There are 3 types of RNA. There is transfer RNA, which copies info into the DNA. There's messenger RNA, which carries the message outside of the nucleus and into the cytoplasm. And there's ribosomal RNA, which actually creates the proteins. ATP, very, very important. Adenosine triphosphate, which is our energy. It's our money. It comes from glucose, which we get from our food, and it creates energy, created into energy by the cells. Glucose is broken into monosaccharides. Energy is released and stored in a molecule, and that molecule is ATP. The burning of the nutrients that are added to the body are used up by the cells through cellular respiration to create ATP. Energy is stored in those phosphate bonds of ATP. Triphosphate, right? ATP is an RNA nucleotide. It's a nitrogen-based adenine with 2 phosphate groups. There are bonds between those phosphate groups that are high energy bonds. And when those bonds are broken, that's when energy is released. To use this energy from ATP, enzymes must move the terminal phosphate group to another molecule. And that's what we call being phosphorylated, which gives it temporary energy to do some work. When the ATP loses a phosphate group, it becomes ADP, adenosine diphosphate. More glucose, more nutrients become metabolized to create phosphate groups, which are then added to AMP, and then which would then create more ATP. So what did we learn today? Biochemical reactions within the body are necessary to understand the structure, the function, the organ systems, and the enzymatic reactions, etc. Everything within the body and how it is made and how it interacts with the environment. That's a lot of stuff. So you must have a lot of questions. I want you to be able to bring those to class. So please write them down. Go back through the lecture, figure out what you still don't understand, and let's make it come alive for you in the classroom.